Hi, my name is Zoe.
Hello, my name is Kim.
Hi, my name is Charlie. Hi, my name is Yakyahun. Hi, my name is Dia. Hi, my name is Sharifah Nawira.
Hi, I'm Ismail. Hi, my name is Eugene. Hi, my name is Pop.
Hi, Femong. Hi, I'm Kristen.
For this video, we are going to talk about compare and contrast processing and modification of rRNA in prokaryotes and eukaryotes. For the similarities, the rRNA genes in both prokaryotes and eukaryotes are transcribed as larger precursors that must be processed to yield rRNAs of mature size. For prokaryotes, free RNAs are transcribed, processed, and assembled into ribosomes in the nucleus, while in eukaryotes, rRNA processing occurs in the cytoplasm. In prokaryotes, the bacterium E. coli has 7 rRNA operons that contain rRNA genes. Transcription of operon yields a 30S precursor, which must be cut up to release 3 rRNAs and 3 tRNAs, while in eukaryotes, the rRNA genes are repeated several hundred times and clustered together in the nucleus of the cell. They are separated by the regions called non-transcribed spaces (NTS), while the transcribed spaces are the regions of the genes that are transcribed as part of the rRNA precursor and then removed in the processing of the precursor to mature rRNA. For prokaryotes, the ribosome is composed of two subunits, a large subunit 50S and a small subunit 30S. The 50S subunit composed of the 20S rRNA, 5S rRNA, and 31 proteins, while the 30S subunit is composed of the 16S rRNA and 21 proteins. While in eukaryotes, the ribosome is composed of two subunits, a large subunit 60S and a small subunit 40S. The 60S subunit is composed of the 28S rRNA, 5.8S rRNA, 5S rRNA, and 50 proteins. The 40S subunit is composed of the 18S rRNA and 33 proteins. For prokaryotes, bacterial rRNA precursors contain tRNA as well as all 3 rRNAs, which are 23S, 16S, and 5S, while for eukaryotes, rRNA precursors contain rRNA including 28S, 18S, and 5.8S without the tRNA. 5S rRNA is synthesized by RNA polymerase III and modified separately. For prokaryotes, RNase3 is the enzyme that performs at least the initial cleavage that separates the individual larger rRNAs. The individual rRNAs are then further processed by secondary cleavage to the final product. Another ribonuclease, RNase E, is responsible for removing the 5S rRNA from the precursor. While for eukaryotes, RNA polymerase I makes a 45S rRNA precursor, which contains the 28S, 18S, and 5.8S rRNA, embedded between the transcript spacer RNA regions. The first step is to cut off the spacer at 5' end of the primary transcript 45S, leaving a 41S intermediate. The next step is involve cleaving of the 41S RNA into two pieces, which are 32S and 20S, that contain the 28S and 18S sequences, respectively. The 32S precursors also retain the 5.8S sequence. Finally, the 32S intermediate is split to yield the mature 28S and 5.8S sequence, which base pair with each other, and the 20S intermediate is trimmed to mature 18S size.
Processing of rRNA in prokaryotes. Prokaryotes have three ribosomal RNAs, which are 23S, 16S, and 5S, which are equivalent to the 28As, 18S, and 5S rRNAs of eukaryotic cells, and are also formed by the processing of a single pre-rRNA transcript. Initial cleavage of pre-rRNA yields separate precursor for the three individual rRNAs. These are then further processed by secondary cleavage to the final product. In prokaryotic cells, each rRNA gene or operon is transcribed into a single RNA precursor that includes 16S, 23S, 5S rRNA, and tRNA sequences along with transcript spaces. The RNA processing then begins before the transcription is complete. During processing reaction, the rRNAs and tRNAs are released as separate molecules. Processing of rRNA of eukaryotes. This is the processing scheme of 45S human rRNA precursor. In step 1, the 5' end of the 45S precursor RNA is removed, yielding the 45S precursor. In step 2, the 41S precursor is cut into two parts, the 20S precursor of the 18S rRNA and the 32S precursor of the 5.8S and 28S rRNAs. In step 3, the 3' end of the 20S precursor is removed, yielding the matured 18S rRNA. In step 4, the 32S precursor is cut to liberate the 5.8S and 28S rRNAs. Lastly, in step 5, the 5.8S and 28S rRNAs associate by base pairing. Unfortunately, in prokaryotes, the rRNA modifications are unknown, but in eukaryotes, small nucleolar RNAs associated with the proteins in small nuclear ribonucleoproteins (snRNPs) required in rRNA processing molecule. The RNA parts of the snRNPs base pair to specific sites within the rRNA precursor and dictate either methylations or pseudoredylations. At those sites, result in modified nucleotides persist in the matured rRNAs that help to define what regions of the precursor to remove and what region to preserve.